First, thank you very much for the very kind invitation. I'm very pleased to be here and to have already had the opportunity to listen to great speakers in the previous session. And I would like to also take the opportunity to congratulate the awardees of the Kyoto Prize this year. So I will give some generalities during my talk, but I will also explain how we came to the transformative genome engineering CRISPR-Cas9 technology. And so I will go in some details about the natural mechanism that bacteria have evolved to defend themselves against their predators, and that is a mechanism that has led to this technology. So it's really, again, as it was said at the previous session, it's a very good example of how a very simple yet sophisticated mechanism in nature can be harnessed as a powerful technology. I would like to start by showing you the first page of an article that was published in the journal Science 3 years ago in the summer 2013 by Elisabeth Penisi, who was already defining this field of CRISPR, the CRISPR craze, with the subtitle, "The Bacterial Immune System Yields a Potentially Revolutionary Genome Editing Technique." And this is indeed about a technology that allows precise surgery on genes and genomes in any cell and organism. And this is very useful for different applications, trying to understand mechanisms of life in different areas of biology. Here I would just like to focus on the fact that surely genetics has been very important as a technology to increase our understanding of diseases at the fundamental level, whether we speak about cancer diseases, heart diseases, genetic diseases, or brain diseases. And it's all about understanding the mechanisms involved at the cellular and molecular levels, always with a wish to understand better the cause of diseases and the mechanisms of life in different types of cells and organisms, always with a wish to identify pathways, genes, and molecules that could be useful for developing novel therapies or novel technologies, whether we speak about preventive or curing therapies, whether we speak about medicine or new types of surgery. And with regard to CRISPR-Cas9, it's really about gene medicine and gene surgery. And actually, you have also a lot of scientists focused on trying to understand infectious diseases. And here I just want to say that actually CRISPR-Cas9 came in my lab from our wish to try to understand the mechanistic background allowing to explain how bacteria cause diseases. And we know by doing such research that we hope to identify pathways that will be important to develop novel anti-infective or novel antibacterial approaches, and also novel technologies with regard to what I'm going to explain you later, with a number of molecular biology tools that actually originate from research on bacteria. So with regard to genetics, there are different important milestones which I would like to mention, so these are just selected ones. And just to remind you that in the 19th century, different rules with regard to fundamental genetics were revealed, such as rules on the origin of species, laws of segregation of alleles, and DNA that was isolated in 1871. 80 years later, the DNA was shown to be the carrier of genetic information. In the 50s, the structure of double helix of DNA was revealed. Then the genetic code in 1966, and surely the wish to understand the genetic code and understand the functions of genes. And you have some selected key events with regard to the basics of genetic tools highlighted here, like in the mid-70s, a lot of efforts trying to identify enzymes and that can cleave DNA or that can allow to recombine DNA. And here, a lot of those enzymes have as origin bacteria or viruses. And a lot of those enzymes were identified trying to understand how bacteria defend themselves against infections by viruses. Surely the enzyme that allow to sequence the DNA, amplify the DNA. We heard about really the efforts of sequencing entire genomes, whether it is these are genomes of prokaryotes or higher organisms such as humans, and a lot of information has been revealed by these genomes, and also the idea to correlate the genome with really the information on those genomes and the codes, the functions of the genes. And you have also RNA interference. That was an important technology developed from trying to understand how regulatory RNAs are involved in the control of gene expression in higher organisms and also lower organisms. And this RNA interference has been used to knock down gene expression in higher organisms. And then two types of nucleases that have been engineered more recently, allowing to perform addition of genes in any type of cells and organisms. And here, those nucleases originate from trying to understand initially the original function of meganucleases and homing nucleases, and that are natural nucleases. And Zinfinger nucleases, Talon nucleases are really engineered nucleases that do exactly what CRISPR-Cas9 can do, meaning that those nucleases can recognize a certain sequence of interest on the genome and act to introduce double-stranded DNA breaks that can trigger modification of the genes, of their sequences. And those tools were working, maybe not so much in a very easy fashion, in the sense that although they can work very well in certain conditions and for certain purposes, still quite time-consuming and difficult to use for someone who's not really, who does not know really how to design this technology. And CRISPR-Cas9 really brings a level of simplicity in the sense that it's a system that relies on a two-component system that is composed of a protein, here indicated in blue, and an RNA component, and that is the programmable component of the system. So in principle, you have the DNA to be targeted, and the enzyme is guided by an RNA component that is in nature two RNAs, CRISPR RNA and tracer RNA, and for the purpose of simplicity, those two natural RNAs are linked together via a linker, and in certain purposes, the two RNAs are used together without the linker, and for other purposes, the two RNAs are used as a single guide RNA. And the idea is that the way the system works is such that one can program the RNA by exchanging the green sequence of the RNA by any sequence of interest to recognize a certain specific sequence on the DNA to be modified. You have another feature in the system that is important to consider, is a so-called PAM, a Protospacer Adjacent Motif, and that is actually composed of a short stretch of nucleotides that has to be present on the genome to be targeted by the system downstream of the guide RNA so that the system can work. And I will explain you exactly how the system really recognizes the DNA. So in principle, it's an easy system. One just needs to reprogram the RNA according to the portion of the genome to be modified. And the enzyme is using two distinct nuclease domains to cleave independently each strand of the targeted DNA. And so the main part of my talk is really to explain you how we came to this system. And we came to this system because in my laboratory we are interested in different bacterial species able to cause diseases in humans. And we focus mainly on bacterium called Streptococcus pyogenes, also known as a group A Streptococcus. So here you see the cocci highlighted colored in red for the purpose of the picture, and that are invading epithelial cells, pharyngeal epithelial cells here, colored in green for the purpose of the picture. So this is really a strict human pathogen responsible for a wide range of diseases. And in the laboratory, we are really interested in understanding the diversity of the clinical isolates for a certain bacterial species, and also to try to understand different types of mechanisms that are using proteins and RNA to allow the production of virulence factors and any other factors that are important for the adaptation of the bacteria in their environment. In this case, is the human host, and also trying to understand how the bacterial pathogens produce virulence factors that can lead to the attack of the human host, knowing that the clinical isolates have always a diversity of factors allowing these attacks. And surely the human host has developed a defense system against the bacterial pathogen, and we work also on this aspect. And this is what mostly is defined as infection biology. So a higher organism infected by bacteria, fungi, or viruses. But you have another definition of infection biology, that is that the bacteria can act as hosts themselves, and they can be infected by their predators that can bring new traits to the bacteria, but that can also be nefast for the bacteria. And these are mobile genetic elements, such as the viruses of the bacteria called bacteriophages and the plasmids. The plasmids can bring new interesting genes to the bacteria, such as genes encoding antibiotic resistance or factors important for the adaptation of the bacteria, or factors of virulence. And the phages can enter two different cycles. A phage can infect a bacterium by invading its own DNA. And the phage can enter the lytic cycle, whereby you have replication of the genome of the phage, production of particles that can kill the bacteria and propagate to kill other bacterial cells. And you have the phages that can enter a lysogenic cycle, whereby they are called temperate phages, and they can, upon insertion of the genome in the bacterial cell, can have the genome of the phage inserted into the genome of the bacteria as prophage, and they can bring new genes, new interesting genes that can be propagated upon cell division of the bacteria. So these are new genes that the bacteria have inherited. And CRISPR-Cas is really one of the mechanisms that the bacteria have evolved to defend themselves against the attack by the mobile genetic elements. And they have different kinds of defense systems that the bacteria are using. I just would like to cite the restriction modification system that has led, as it was mentioned in the previous session, to the identification of restriction enzymes that have really also revolutionized molecular biology. And this is really by trying to understand these restriction modification systems in bacteria that those enzymes were identified. And the CRISPR-Cas is also a system, a natural system of defense, and by understanding the mechanism of this system, we have developed this CRISPR-Cas9 technology. What is important to mention is that all the defense systems existing in bacteria are considered as innate immune systems, in analogy to the adaptive and immune systems known in higher organisms. And CRISPR-Cas is an RNA-mediated system and is considered as the first and only adaptive immune system. It exists in bacteria and archaea. It's not present in all bacteria and archaea. It's only present in 50% of the bacterial species, the complete genome of which has been totally sequenced. And 90% of the archaeal species is just an accessory defense system. As I said, there are other defense systems existing. And for those who wonder what CRISPR means, it means clustered regularly interspaced short palindromic repeats, and Cas means CRISPR associated. And you will understand what I mean by repeats and short palindromic. So this is how the system works. You have three phases in the system. You have a phase of memorization, whereby the bacteria is going to memorize a viral infection, then an expression phase where all the components of the systems are expressed, and then an interference step whereby the components CRISPR-Cas can recognize the phage upon a second infection. And so you have the so-called CRISPR-Cas locus, and the locus is composed of a CRISPR array that is itself composed of a region in the DNA that can contain a promoter and that allows the expression of this so-called CRISPR array into the RNA component of the system. And the CRISPR array is itself composed of very short repeats that are small sequences identical to one another and that are interspaced by sequences here highlighted in green that are called spacer sequences and that are actually unique on the genome, and they are different from one another. So this CRISPR array really is expressed as the RNA component. And then you have a series of genes, the CRISPR-associated genes, organized as an operon. And this operon encodes the CRISPR-associated protein. So this is the protein part of the system. So you have a protein, you have the protein components and the RNA components. And so you have a first phase of so-called memorization, and it's called spacer acquisition. So the spacers have as origins either plasmid, transposon, or phage origins, so the origins as mobile genetic elements. And what will happen is when a phage, for example, infects the bacterium that contains the CRISPR-Cas system, some CRISPR-associated proteins will recognize the invading DNA of the phage, will cleave a portion of the invading DNA of the phage, and will insert it into the CRISPR array at the level of the leader sequence. And this works as a kind of way to memorize the phage infection by inserting a portion of the phage into the own genome of the bacterium at the level of the CRISPR array. And then you have expression of the system, whereby the CRISPR array is transcribed into a long RNA molecule that is cleaved together with a protein encoded by the Cas operon that works as an endoribonuclease, and that will recognize the repeat sequences that in this case can form palindromic hairpin structures. And the protein, the endoribonuclease encoded by the Cas operon, recognizes these structures and can cleave the RNA to produce mature RNAs that are each individual, distinct from one another, and each of the RNA contains a specific sequence of the specific phage or plasmid and transposon. And these RNAs can guide a complex of CRISPR-associated proteins encoded by the Cas operon to the invading genome upon a second infection of the phage. And here there is a recognition based on the base pair complementarity between the green sequence of the guide RNA and the corresponding green sequence on the phage. And when there is recognition, one of the CRISPR-associated proteins can cleave the invading genome. So this was a dogma for the CRISPR-Cas systems when we started to work on the system. And there is a large diversity of the system, like that corresponds to the large evolution and diversity that you can find in the macrobial world, as it was also mentioned by the previous speakers. So here, the diversity of the system is based on mainly on the nature of the genes encoding the CRISPR-associated proteins. So you have two classes of CRISPR-Cas systems that now have been classified. And you can see that the class 1 system contains a large number of CRISPR-associated genes, whether the class 2 system is more minimal and contains, for example, for the interference step, that is in orange-yellow, only one gene, whereas you have a series of genes for the type 1 and the type 3 systems. And this is what I referred to here when I said that, in principle, the dogma was that you have one RNA molecule containing the memorized mobile genetic element to be targeted and a complex of CRISPR-associated proteins working together to really work to mature the RNA and to target the invading genome. There is a large history on the system, so I will mention only key points. The first identification of the CRISPR-Cas locus was made by a group in Japan in 1987, showing the presence of these repeat sequences in the genome of E. coli. not knowing really the function of those repeats. And then you have different scientists who actually found out that those repeats were interspaced by sequences and could be transcribed as RNA species of different sizes. Then the identification of those sequences of mobile genetic element origin. Then the fact that this CRISPR array was located in the vicinity of this operon that encodes genes that were clearly, is composed of genes that were clearly encoding proteins that can interact with DNA and RNA and cleave DNA and RNA, and with the hypothesis by different bioinformaticians, including the group of Eugenie Konin at NCBI, that actually the system could work as an RNA-mediated interference system by analogy to the RNA interference systems existing in eukaryotes that are using macroRNAs and small interfering RNAs. And then different biologists really look into the biology of the system. There was a paper published in Science in 2007 showing that actually the system is indeed an adaptive immune system, and there is a first recognition of the system to then lead to immunity, and this was done in Streptococcus thermophilus with the first application in biotechnology, and that is to repurpose the system in a natural way to produce Streptococcus thermophilus strains that can be naturally engineered to be resistant to viruses and that are used for production of milk products, where the threat is always the killing by phages that one can find around of these bacteria that are used for the production of milk products. And then different scientists who really studied the type 1 and the type 3 systems and came to this idea that you have an RNA molecule guided by a complex of CRISPR-associated proteins to target the genome. So you have different names I can mention with a group of Philippe Horvath, Sylvain Moineau, Stan Browns, John van der Ost, and Michael Turns, and Luciano Marafini, and Eric Santimer, and other scientists who really worked and did very nice work on those systems. In my lab, we focused on the type 2 system, I would say thanks to Streptococcus pyogenes, that is a bacterium that contains at least the clinical isolate that we have studied, the type 2 system, and also one of the type 1 system. But the system that was active in our strain was a type 2 system. And we came to work on CRISPR-Cas9 because we were interested in the family of small regulatory RNAs among the different mechanisms that we tried to study in the laboratory. So we focus really on the regulation of gene expression. And small regulatory RNAs are important components in the regulation of gene expression, whereby RNA is known as a molecule like the messenger RNA that works as a transition molecule between the DNA and the proteins that are synthesized, and you have also the ribosomal RNA transfer RNAs that are important for translation. But the small regulatory RNAs, so in bacteria they are called small regulatory RNAs, in eukaryotes they are called small interfering RNAs, macroRNAs, and other types of RNAs with different names. The small regulatory RNAs in bacteria, they can modulate gene expression by targeting directly messenger RNA via antisense mechanisms, and they can change the stability of messenger RNAs, or they can also activate or inhibit translation, but they can also directly target proteins and activate or sequester them from their original functions. There was a missing link when we started to work on CRISPR, that was a small regulatory RNA that will target directly DNA and affect somehow DNA in some ways. What was also missing is a regulation whereby a small regulatory RNA would be important to activate the function of another small regulatory RNA, and these are two aspects in fundamental mechanisms of RNA-mediated regulation that have been brought with CRISPR-Cas9. So about 10 years ago, we were interested in understanding more about regulatory RNAs. We were focusing on a couple of those regulatory RNAs, but we used the bioinformatic approach, and this led to the highlighting of a number of regulatory RNAs. Here are only those at the time which we could show where RNAs well transcribed and which would have a potential interesting role in the regulation of gene expression. And we focused on this RNA called pRNA-49, now known as tracer RNA. And this RNA was of interest for us because it was well transcribed. So here's a Northern blot analysis showing the transcription of this RNA in the bacteria, and this RNA is well transcribed throughout the growth phase. We always look at the expression throughout the life of the bacteria from early log phase to late stationary phase. So this was transcribed into 3 species. We also mapped the start and the stop of this RNA molecule. What was of interest for us is that this RNA was located was encoded in the vicinity of a gene that was predicted at the time to be a CRISPR system-related protein that contains 2 nuclease domains, RUFC and HNH-like nuclease domain, from Streptococcus thermophilus. So here we work with Streptococcus pyogenes, and this is actually now what we know as being the gene encoding the Cas9 protein. In principle, we were working on this tracer RNA molecule, and we had found a messenger RNA, which this RNA could target, and this was a messenger RNA encoding a virulence factor, and we were trying to show that tracer RNA had an effect on the expression of this messenger RNA and could somehow control the expression of a virulence factor. And although we had a nice interaction of antisense, so tracer RNA, messenger RNA, we could not make really any sense of this function, and I think we know why. But in principle, trying to really struggle with gaining a function for tracer RNA, it was clear that this tracer RNA was located in the upstream, in this case for Streptococcus pyogenes, upstream of the type 2 CRISPR-Cas locus that is defined as this gene encoding the Cas9 protein, and that is a minimal CRISPR-Cas system. And the idea that I had was to look whether by any chance tracer RNA will not have a function that will be important for the activity of the system and the functionality of the system. And here I don't show you the details, but we used a thorough genetic and biochemical approach, and actually for this study to really understand the mechanism of the type 2 system, we really used genetics with tools that I developed during my time in the US that really allows to perform precise gene surgery on bacteria. And in principle, we showed by doing a number of experiments this model, whereby tracer RNA is an RNA that contains in its sequence an anti-repeat region that allows the RNA to base pair with each of the repeat of the long RNA molecule to form a duplex of RNAs that in this case is stabilized by the protein Cas9. So Cas9 can recognize this dual RNA structure and allows to stabilize this duplex formation between the CRISPR RNA and the tracer RNA. And you have a recognition by an endoribonuclease from the bacteria, RNA3, that can recognize this dual RNA structure, can cleave the dual RNA structure to lead to the formation of these intermediate forms of CRISPR RNAs, still bound to tracer RNA, with processing by unknown exo or endoribonuclease to lead to the mature form of the RNA. I told you that there is always production of single mature RNAs, and in this case, it is composed of the green sequence that can target the phage genome and a portion of the repeat that can still form a duplex with a tracer RNA and stabilized by the Cas9 protein. So when we came to this step in principle, the prediction that we had in my lab was really that this was not a dead end for tracer RNA, in the sense that tracer RNA was not only involved to really trigger the maturation via cleavage by RNA3, but will be important to work as really a component essential for the interference complex. I should say also here that the involvement of RNA3 in the mechanism was interesting because it was reminiscent to mechanisms that are involved to to produce small interfering RNAs and macroRNAs in eukaryotes for RNA interference, and that rely on dicer and Rocha enzymes that have also RNase-3-like functions. Having said this, this was clear that it was also a mechanism that was distinct from the type 1 and type 3 mechanisms, in the sense that here you have one protein and two RNAs instead of one RNA and a complex of proteins. So in principle, the goal was really to show that this is the complex that is doing the work to really recognize the phage genome and cleave it. And because we were intending to do biochemistry to show this, and also to try to see what is really the minimal portion of this system doing the job. The idea was really to show that indeed the system was really active in Streptococcus pyogenes. And when we looked at the nature of the spacer sequences, we figured out that these sequences can target phages. And in principle, this system in Streptococcus pyogenes, it's a defense system against phages that can bring new virulence genes to the bacteria so that they can cause more severe diseases. And to really show that the system is active, we did some phage experiments, and we also developed a readout system using plasmids, whereby we could show that indeed when we have a wild-type Streptococcus pyogenes having the CRISPR-Cas system here that is active, the phage genome will invade the bacteria, will be recognized by the CRISPR-Cas system that will cleave the genome, and you will have destruction of the phage. And this will mean that you don't have phage DNA lysogenization, meaning the phage DNA cannot be incorporated into the genome of the bacteria. And you cannot have acquisition of virulence genes, because this is the genes that are carried by these phages. And if you knock out CRISPR RNA, if you knock out the other RNA tracer RNA, if you knock out the gene encoding Cas9 protein, and if you knock out the gene encoding these RNAs that is important to produce the mature forms of the RNA, you have a dead system. And in this case, the phage DNA can enter into the phage DNA invades the bacterial cell and can be inserted into the genome of the bacteria, and you can have phage DNA lysogenization. So we did the experiments to show that the system was working as such, and that those components were important to target the phage genome. And here I just show you an important experiment that is the most important experiment that says everything, and it's really to show that this complex is important to target the DNA. So here you have the phage DNA that is targeted by the system. You have the PAM sequence, that is a short stretch of nucleotides composed of NGG here for this specific purpose. We have two possible NGG in this specific example. And you have the mature CRISPR RNA composed of the green sequence that can base pair with the target phage DNA. You have the repeat sequence that can base pair with tracer RNA. And this PAM sequence is important. It allows to recognize self and non-self. And you have some bioinformaticians who have identified this sequence. And biologically speaking, it has been well studied by the group of Sylvain Moineau in Quebec. And he had published a paper in Nature showing that actually the CRISPR-Cas9 system could recognize DNA via this PAM sequence that was important and could cleave the DNA, although at that time tracer RNA was not identified and it was not clear how the interference system would work. And here is just a simple experiment. If you want to show that indeed this is a complex doing the work, the idea is really to purify the different components, the protein, the two RNAs, having the target DNA in the test tube and do a very simple cleavage reaction on the DNA. And here we worked on DNA that had a circular form and linearized form, and we could see cleavage on the DNA. So this is a gel showing the different bands of the DNA, and we could observe cleavage when we had indeed this tracer RNA introduced into the test tube. If we don't have tracer RNA, the system does not work, and it's really a dual RNA guiding Cas9 to cleave the DNA in a sequence-specific manner, cleaving the DNA 3 base pairs upstream of this so-called PAM sequence. And here you have two possible NGGs, so you have two possible cleavages, and the cleavage is blunt. And the way it works is that the enzyme has a domain that allows to recognize this PAM sequence on the DNA. So it first screens the genome to find this PAM sequence. And then there is an homology search, whereby the system will try to recognize a sequence that is homologous to the CRISPR RNA. And you have opening of the DNA then when you have recognition, so it's what we call R-loop formation. And you have the enzyme that will be able to cleave the DNA using 2 nuclease domains. And each time you have a change of structure of the protein that changes always its structure when it's binding the RNA, when it's find the DNA, and when you have homology between the RNA and the target DNA, and also a repositioning so that the enzyme can cleave the DNA. And you have double-stranded DNA break that is occurring. So it's really the understanding these pathways that we came to this system. And the system naturally is using two RNAs, and the system is working very efficiently and really using the natural protein. And so the idea was just to simplify the system and to just add a linker. So in principle, the single guide RNA is basically really the natural system. Having said this, so this is really how it works, and surely to adapt it to higher organisms, what the manipulator has to do is to add a nuclear localization domain to the protein Cas9, because indeed it's a bacterial protein and there is no nucleus in the bacterium. So one has to fuse Cas9 with to a nuclear localization domain, and to change the codon usage of the protein so that it can be well expressed in human cells, and then to express an RNA that contains a sequence that can target a certain sequence on the genome of interest, as long as it is upstream of this NGG, but it's a restriction, but you have a lot of NGGs on the genome. Having said this is the only thing to do, and the system was working so well that the community could really see that the system was working in any cell and organism that were tested, basically using the system, just changing the codon usage. And so it's a very efficient system. And one has to say that the system that is used worldwide is really the system originating from Streptococcus pyogenes. But the system has largely evolved in bacteria, in the sense that even the type 2 CRISPR-Cas9 system is largely evolving. And when you compare the sequences of the proteins and the repeats of the RNA and the tracer RNA in one species versus another species, you see a large variation of sequences. So the proteins have some conserved domains that allow to cleave the DNA, but otherwise the sequences are largely diverse, and you have also a diversity of PAM sequences, meaning that all the systems existing in bacterial species have evolved to recognize different motifs on the DNA. So this allows to enlarge the toolbox by having different CRISPR-Cas9 systems. So what CRISPR-Cas9 is doing, it's exactly what zinc finger nucleases and talon nucleases are doing, except that they use a different modes of recognition. They use modes of recognition of the DNA that rely on a code that is, that is included in the protein. So in principle, for zinc finger nucleases and talon nucleases, one has always to engineer a new protein for a certain sequence on the DNA to be recognized and cleaved. For CRISPR-Cas9, it's just the RNA component to program, and it's easier. And so when you introduce a double-stranded DNA break on the DNA, and this is what those enzymes are doing, they just trigger a break, the rest is done by the cell. And in eukaryotic organisms, which is actually not really the case for prokaryotic organisms, you have non-homologous end joining repair mechanisms that are highly effective, at least more effective than those repair systems in bacteria. And when you use these systems that are triggered upon breaking of the DNA, you can introduce small or larger indels on the genomes, meaning you can delete smaller or larger portions of sequences on a genome in a sequence-specific manner. And if you introduce an homology donor, you can replace a certain sequence of interest by another sequence of interest. So you can modify a mutation, you can introduce a mutation, you can introduce a new sequence at the site of interest. What we had predicted was actually that the system could offer considerable potential for genome editing in cells of the three kingdoms of life for biotechnological, biomedical, and gene therapeutic purposes. And also an idea that I had that maybe it will be useful for developing the technology to treat human severe genetic disorders. So the system has certainly advantages, which everyone agrees with those advantages. It's cheap and easy to use. It's very efficient. It's working very well if it's delivered. It's also versatile, and I will explain you why. It allows multiplexing in the sense that by designing different programmable RNAs, one can at once target different portions of the genomes at once, and it can be developed into various flavors. It's highly specific, even though for certain aspects, sometimes you have the problem of off-target effects, and you have the system that has already been developed substantially to increase the specificity. It has low toxicity, and the challenges for the future are really to develop this delivery system that allow to bring the technology into the cells and organisms in an efficient manner. So very fast, about 7 months after we published the last step of the mechanism of the system and how to engineer it, scientists found that indeed it could be harnessed as a tool to manipulate the genome of bacteria, plants, mice, human cells, later on organoids and monkeys, and also zebrafish, Drosophila. So it's very important for all this organismal biology, and to try to really increase the knowledge of the functions of genes. So you have substantial work that has been done on human cells, and a lot of engineering with the implication of colleagues from the Boston area with the group of Fang Zhang, Keith Young, and George Church, and then a lot of scientists who have joined and done really excellent work since. So these are some aspects of the Cas9 toolbox. As I said, you can target directly the DNA, and you can introduce larger or smaller deletions. You can insert new sequence, replace sequences, introduce mutations, correct mutations. But the nice aspect of the system is that naturally it has 2 nuclease domains to cleave. And so you can mutate one nuclease domain and transform the protein into a protein that is no longer cleaving the 2 DNA, so the 2 strands of the DNA, so working as a nuclease, but you can knock out one domain and have an nicase, an enzyme that will be RNA programmable to cleave one strand of the DNA. And you can also use the technology to really, transform the system to somehow target a nuclease that will have the 2 domains knocked out to have an RNA programmable DNA binding protein, whereby you can fuse to this DNA binding protein that cannot cleave any longer the DNA, different domains that allow to activate gene expression or also downregulate gene expression. So you can really act on the expression of the RNA of the system by having an RNA programmable enzyme, that actually not enzyme, an RNA programmable DNA binding protein that can activate or inhibit transcription in a sequence-specific manner. And you have a lot of screens that have been developed in this aspect. And you can also fuse to this dead Cas9 that is RNA programmable. You can fuse an effector domain, such as a domain that allows to change the epigenetic marks on the DNA and understand the epigenetic phenomenon. And you can also fuse to this dead Cas9 a domain that allows to mark the DNA in a sequence-specific manner. So you can also really, boost these technologies that allows to do imaging location of the genetic locus. And the technology is developed week after week to always bring new aspects. So I would like here to speak specifically about what I told you. So the system system type 2, that is a CRISPR-Cas9 system. Actually, this Cas9 enzyme had different names prior to Cas9, CSN1 and Cas5. So there are some publications referring to Cas5, CSN1, and actually it's Cas9. But in principle, when we looked at the genomes of the bacteria, when we understood how tracer RNA could work, right away we searched whether we could find an homologous molecule of tracer RNA in different bacterial species, and we found that indeed all the type 2 systems are always associated to this tracer RNA molecule. and that you can find this type 2 system in more than 350 bacterial species. And so you can do some phylogeny. And as I said, you have a large diversity of sequences of the genes encoding Cas9 and also these repeat sequences and tracer RNA molecule. And you can somehow understand the coevolution. And very fast, what we figured out is that the repeat sequences of the RNAs are always different in terms of nature of sequences when you compare the different bacterial species having the type 2 system. However, you have a coevolution whereby you have always a conservation of this duplex RNA formed between tracer RNA and CRISPR RNA, even though the sequences are evolving. And you can also classify the type 2 system into 3 subtypes based on the sequences of the Cas9 proteins. And you also can correlate the evolution of the Cas9 proteins with the architecture of the CRISPR-Cas locus. And you can also define that if you look at different phylogenetic groups, so if you group the Cas9 via specific groups, you can also correlate the sequences of the Cas9 protein with a certain architecture, a certain structure of this dual RNA structure. And here I just show you a diagram that shows that if you have the protein of a phylogenetic group that has a green color, it can work together with the dual RNAs of a species of the same phylogenetic group, but it will not work together with the RNAs of another phylogenetic group. So what I want to say by this system is that you have some conservation, you have some diversity. And this is really by understanding the diversities and the conservation of the system that in my lab we really understood how it works. And those, how the systems really work together, how the different components can assemble together in terms of the phylogeny are important to really develop the systems of other bacterial species, knowing that so far a lot of bacterial species have been tested, so CRISPR-Cas9 from different bacterial species. Everyone agrees to say that the one of Asparagines is really the one working the best. Some systems from other bacterial species do not work very well in higher organisms, with really also the idea to work with a smaller Cas9 protein and the largest Cas9 protein to try to understand the minimal portion of the system that is important for the system to work, and also working with minimal components of the system to have them more amenable when one wants to deliver a genetic tool. It's always important to work with a short system. Here I just want to point out the applications of CRISPR-Cas9 in biology. So it's really transformative in life sciences to understand the functions of genes, to lead to the production of new crops in agriculture, for synthetic biology to engineer new pathways, also for gene drives and for human medicine, with different aspects that is surely it boosts having genetic tools that is easy to use, where one can control, gene expression in a versatile manner, and one can also do precise genetics. One can understand better the functions of genes and unravel new pathways. It's very important to have these genetic tools to engineer disease models that are more close to the clinical situation, and animal models and humanized animal models. It's very useful for really developing these screens to find new targets for therapeutics, to also validate new therapeutic targets and medicine under development. It's important for the bed-to-bench approach with what was mentioned in the previous session, with really the increasing understanding of these genomes of human patients and humans in general, where a number of mutations are found on a regular basis, whereby those mutations are thought to be predisposition for certain types of diseases, and one can really go back to these models and test indeed that the mutations may be causative for certain types of diseases. And the challenge that is really to develop the technology to develop for direct therapeutics to treat human genetic disorders, whether one combines gene therapy with cell therapy in an ex vivo approach, whereby the cells are collected from the patients and somehow corrected and reimplanted in the patients, or whether it's an in vivo approach when the technology is directly targeted to really cure the patient directly. And I will just finish by saying that you have other simplified systems that are now studied. And we studied recently the CPF1 system. And here it's a system, and I'm just going to show you three slides, whereby the protein CPF1 can mature the RNA and can also target the invading DNA. And this is how it works. It's in another bacterial pathogen for humans, Francisella novicida. You have the RNA that is produced. You have the enzyme that contains within one enzyme a domain that can recognize the RNA. So it has an RNase domain that can cleave the RNAs. When the mature RNAs are produced here, they can guide this protein here to the invading DNA and cleave the DNA not in a blunt fashion, but by producing 5 nucleotide 5' overhangs. And so this is another type of systems that is existing. And others and us as well are, like the group of Fang Zhang has revealed already functions of some of the CRISPR-associated proteins belonging to the class 2 minimal system. And we are also focusing on trying to understand those steps of memorization, because this involves proteins that also recognize DNA and insert recombined DNA into the genome. So we really hope that this will lead to new mechanisms, and also increased research on bacteria will lead to really, deciphering mechanisms that could be exploited for new tools for genome engineering. So in my case, really the study developed mainly in Sweden at Umeå University, but based on research that was done at the University of Vienna initially. And I have to thank different institutions in Germany, so the Helmholtz Center for Infection Research and Max Planck Institute for Infection Biology. Wonderful PhDs and postdocs who have participated to the work for CRISPR-Cas9, essentially Christoph Schilinski, Elisa Delcheva, and this is a team for CPF1 with Ines von Verhagen, Richter, Maida Bratovic, and Anaïs Lerin. And collaborations, the group of Jörvogel for RNA sequencing analysis that was important for the first step of the system. Then the group of Jennifer Donna with Martin Jinek, with whom we have done corresponding quite a lot to establish the structure of the system and the biochemistry with DNA interference. And the group of Eugenie Kounin for the evolution part. And I need to mention for conflict of interest, ERS Genomics and CRISPR Therapeutics, that are two companies that I have co-founded. Thank you very much for your attention.